I see people are here. Hello. Looks like Susan's here. Well, anyways, I don't think we're going to have a full meeting today. It's pretty early in the year. I see a couple people here who want to, maybe they want to introduce themselves. And I know maybe some of you were in the Slack channel for DevoWorm, so that's good. If you're interested in the group, join the channel. We have meetings here, usually on Mondays, sometimes we don't. And then we have our YouTube channel where we have all sorts of things related to the project. So looks like we have Shreyas and Varshit. So would you like to introduce yourselves?
Myself Shreyas. Me joining for the first time. So I got interested in this and I was looking up the repo. So yeah, like this is my first meeting. I'm joining for the first time.
Okay.
Yeah, recently, like I've been working in deep learning. I'm not from a neuroscience background per se, but I recently have been like interested in applications of deep learning, deep learning in neuroscience. So yeah, kind of want to know how it is and interested in this field. Thank you for having me.
Yeah. So yeah, that's something we of course we've done a lot of with small connectomes and with cells and things like that. So yeah, that's something. I don't know if you're looking to build a project. We should talk about that though. To come up with ideas. We have data sets available. If you look at the things we were doing in the last Google Summer of Code, I think you can get an idea of what we are interested in. And those videos are on YouTube. If you go back to like last summer in some of those meetings, or look at the repo and see what kinds of repositories we have. So I know you can't learn everything from the documentation, but if you have questions, that should guide you to those questions, and then we can discuss that.
Yeah, sure. So primarily, I got to know about this organization through Google Summer of Code itself, to INCF, and then I looked up the repo and the projects for past year, the hypergraph project, and it looked pretty interesting.
Yeah, we've been doing the hypergraph project for probably 5 years now. And just various versions of it and building on what we have. So that of course is in the DevoWorm organization. So, and again, we basically the hypergraph project is where we have data like image data, secondary data of different types. We use some sort of hypergraph algorithm to analyze them, and we've had different ways of thinking about this. Last summer we had a project where we really kind of focused on the representation. The summer before that we worked on some implementation, so all the code is in there that has been developed. The question is, what can we do with it or how can we make it better? Because I don't want people to keep reinventing the wheel, obviously. So look through that and see what you think, what you can contribute or what you find interesting.
Yeah, I'll be looking at like the last year's project and understanding what was the progress done, and then I will ask you questions regarding it.
Okay, sounds good.
Yeah.
Thanks. I don't know if Varshit is available. Is that a note taker app or is that?
This one seems to be the note taker app, I guess.
Okay. Well. All right. So it's good. Susan, hi.
Hello. You started late today, or?
No, I'm just checking in to see what people are up to.
Oh, okay. What am I up to? Collecting papers. I need to start reading.
Yeah.
I have to write a paper about my thesis, so I could present it to the group at some point if I could get going.
Yeah. Yeah, I don't know what. Well, it's a pretty big, tall order paper based on your thesis. Like, because you could have a lot. It could go in a lot of different directions.
Yeah. I think I have to compare regular mechanics with what I'm doing.
Okay.
So what have I got here? Yeah, mechanics and biomimicking of wet adhesion in nature. There you go.
Okay.
Talk about a large paper.
How long is that?
44 pages.
Okay, well that's not bad actually.
Yeah, it could be worse.
Yeah, it's a nice review.
Anyway, I'll look at it. I just have to compare continuum mechanics to tensegrity. So
Okay.
I need to do some searches and yeah, there's been a few articles. I've got quite a few. So including optical coherence elastography. A review of that. So
Yeah.
and I've got this one might be interesting. Constitutive framework for tension, compression, failure, asymmetry in soft materials.
Okay.
I don't know. Anyways, whatever. I've got things I have to review here, so I guess it's better than going for a walk out in 30 below. So we'll you know.
Yeah. Well, that's good. Yeah. So yeah, keep us posted on that. That'd be good to see progress on that.
And by the way, before Christmas, the last paper you presented, I did find it interesting.
Which one was that? I can't remember.
On the neurons, they're growing them in gels.
Oh yeah, the paper that is
Patterns.
from the Mind in Vitro group that Morgan is always talking about. So yeah, that's and that's just like growing that on some sort of substrate and getting it to culture. So like in cell culture, you always want to find a good substrate for 2-dimensional, 3-dimensional arrays of things. Like for a simple cell culture, you have a monolayer and you can grow cells out on that. But of course, if you want to understand tissues, you need to have the 3-dimensional layer, and you can build a structure there, and then try to replicate that complexity. And then that's kind of how that people think about that a lot. How to do that on different substrates, how to make sure that they adhere properly, that they grow properly, because sometimes if you get the wrong substrate, you can kill the cells off, or they don't behave in the way that they would behave in the body. And then, of course, they're looking at like things like transplantation and other down the road things. So yeah, it's definitely, I think, really interesting from that standpoint, but also how cells are organized in different populations and different functional populations and that. So
Yeah, there's a method of stiffening gels, or manipulating the stiffness of gels too. So they might be onto something.
yeah. Is it like an electro?
Yeah, add a bit of, well, I don't know. You can put magnetic spheres in it, but also give it electrical stimulation. So there's a couple of methods out there. I haven't studied them. I collected the papers, said that's interesting, and moved on. I think I call that thesis fever or something.
Well, I think you just coined a new term there.
Yeah. Oh,
Thesis
well.
fever. Well, I think that's going to be pretty popular, actually.
Oh, that looks interesting. I can't even look at it. Yeah. Oh, well. anything else? I don't know. If I found a couple of papers on networks of cells. If I can find them, would you like a couple of them?
Yeah, that would be good.
Okay. I don't think I buried them too far.
Yeah. Well, yeah, I look forward to reading that. Always interesting. All right. Do you have anything else you want to talk about?
No, I just trying to do Christmas or New Year's season, and yeah. So
Yeah.
Like since you guys are here, like just as a beginner to understand stuff, like what do you guys exactly research on, and like I look forward to joining the meets and understanding this in more in detail. So since we are having a meet, I would go back and further look into GitHub repos and everything. But just as a starters, like what is the research going on, and what would you suggest to me? Like what should I start with? What concepts and all?
Well, I think probably the Summer of Code stuff. The repository that we have for
Right.
hypergraphs. If you go to our YouTube channel, we have things from, if you search that channel for like hypergraphs, there are presentations that people have given on it. We have reviewed papers on it, and that, going over that and looking at how we talk about it, because we have specific interest in sort of, using developmental biological data for this. So looking at that, going through that, and then thinking about some of the people who were involved in last year's hypergraph project and the year before. You might get in touch with those people as well.
Oh, right. Sure. Who are those people? I would like just get in touch with them. Those who are with this.
So like Leilith Baru was doing the work last year on hypergraphs and neural developmental programs, and Zhai was doing the work on temporal graphs, and so people like that, you'll see them in the GitHub repository. So if you see some code you're interested in, look at that person, and yeah.
Sure. So that project is running, right? So that's an active repo and that's an active project.
Yeah.
Which work, right?
Yeah, it's active.
Okay, nice to hear. Like, looking forward to contributing and helping. Yeah, learning.
Yeah, sounds good. Yeah, thank you.
Thank you. Yeah, and happy New Year.
All right. Yeah, happy New Year. I'm gonna get going. All right. Thank you.
Oh, well, happy New Year too.
Yeah, happy New Year.
Although I think my family have stopped barging into my life here.
Oh, good.
They're all back to work today.
Yeah.
Well, I'm still interested in what Dick wanted with the tensegrity information, but he isn't here, so I don't know if he's looked at it.
Oh, did you send him something on it, or?
Yeah, I sent you and him a dump of about 10 papers.
Yeah, I did see that.
Yeah.
There was...
Have you had a chance to look at it at all?
A little bit, but I haven't really, I don't have anything up and ready to look at.
Oh, okay. Well, I'm like I said, I'm interested in knowing the background math so I can study that before trying to read these things, because I've already tried to read them. I know it contains linear algebra, that's fine. But there's something else going on.
Yeah. I'll make an assessment of it and see what I think.
Okay.
Yeah. All right.
Yeah. Thanks.
All right. You're welcome. All right. Talk to you later. So I wanted to get into a few papers here today. So the first paper is called Non-Darwinian Molecular Biology, and this is by Alexander Palazzo and Kejiou, and they're from the Department of Biochemistry at the University of Toronto. And their argument is, basically that we can't understand molecular biology without thinking about something called molecular neutral theory. And molecular neutral theory has an interesting history with respect to evolutionary theory, and that is it introduces this non-Darwinian force of evolution, which is neutral or neutral drift. So we already think of evolution as having four forces. We have mutation, we have selection, we have drift, and we have recombination. And so drift is already one of those forces. But of course, in neutral theory, we develop sort of a theory of drift and something that operates at the molecular level, which allows for things to drift basically out of function. So we're in and out of function. So if you have a population of organisms, that population of organisms is sort of a sampling of maybe a larger population of organisms, or it's a sampling of the genetic diversity inherent in life or in some phylogeny or some lineage in the phylogeny. And so you're dealing with a subsampling of these things, and you have, because of that subsampling, you end up with these frequencies of alleles that are maybe not representative of Hardy-Weinberg equilibrium or some sort of static equilibrium that you would find in the larger population. So that means that you can have all sorts of changes in evolutionary changes that are not subject to selection but subject to drift. And so in molecular biology, there's been this whole theory of neutrality. This comes from the work of Muthu Kimura in the 1960s, and they've developed that from there. So it's very molecular, and it's basically, if you go to the biochemistry literature, it's almost exclusively a biochemical sort of phenomenon. They talk about it in terms of biochemistry exclusively, but it has an effect on the phenotype because obviously the biochemistry constrains the phenotype in that way. So this paper is a nice review of neutral theory, but it's kind of thinking about the field of biochemistry in general and how, basically they're thinking their words, they call it a drift because they're not thinking about the questions that they're asking in light of neutral theory. And so this is this person, Alexander Palazzo, is at the University of Toronto. And if you go to the Sandwalk blog, which is a good blog if you're interested in finding out things about the theory of evolution and molecular evolution in particular, and looking at some sophisticated arguments for it. Larry Moran, who is actually used to be at the University of Toronto, their Mississauga campus, he he's covered this historically over like the last 20 years. And so this is a post from 2015 on functional RNAs. So in this paper, they mention the distinction between function and non-function. So one of the things about neutral theory is it predicts that most of our genome is not functional. And there have been these recent studies, such as ENCODE and other studies that have proposed that the genome is largely functional, that you can calculate up to 80 or 90% function for a genome, or any genome for that matter. And what they're arguing is that that's not the correct way to look at it, that a lot of these estimates of function are picking up spurious associations of transcription factors with binding sites. And so we need to think about function. When we talk about function, we need to think about it in terms of what neutral theory predicts. And so that's been a thing that Larry Moran has talked about a lot in his blog. is this notion that junk DNA is necessary and, of course, prevalent in genomes. Most of our genomes are junk, and the reason is because this is predicted by neutral theory. It's predicted by the evolution of genomes, the things that have gone into the evolution of genomes. So our genomes are, of course, inherited from ancestral forms. We have recombination events. We have different types of mutational events. We have retroviruses that get incorporated in the genome. And all these things can render genes non-functional. We have gene duplication events, which can expand the number of genes, and some of those can evolve to some sort of non-function. So there are all sorts of events that lead to sort of non-functional components to our genome. And so we should expect to see a lot of non-functional DNA. And so this is a post that he did on functional RNAs and thinking about this same problem of DNA function in terms of RNAs. And so this is kind of going through, and he does cite a paper in this post, Palazzo and Lee, 2015, non-coding RNA. So this is a different paper from this paper. This is non-coding RNA, what is functional and what is junk. And so this is a good paper on this distinction and thinking about this in terms of evolutionary theory and neutral theory more specifically. So if you know anything about RNAs, you know that there's like this zoo of different types of RNAs. You have, there are all sorts of classifications of RNAs, long non-coding RNAs, intergenic RNAs, of course, messenger RNAs, mRNAs, short RNAs, all sorts of things that we can think of. And so there's all sorts of different classes of RNAs. They're all produced from genes being regulated in different ways. And they all, maybe serve these putative functions. But of course, it's kind of hard to know. People name an type of RNA, how many of those are functional, if they're even functional in the way that we would like to think of function. So it depends on our definition of function. It depends on what it's doing across the genome, these classes of RNAs, et cetera, et cetera. So this paper is on non-coding RNA, what is functional, what is junk. This paper is, so that paper was from 2015. This paper was from 2022, and this is called Non-Darwinian Molecular Biology. So this is a larger review of this idea of neutral theory. So why don't we go through the abstract and we'll talk about maybe what are some of the implications of this. With the discovery of the double helical structure of DNA, a shift occurred in how biologists investigated questions surrounding cellular processes such as protein synthesis. So instead of viewing biological activity through the lens of chemical reactions, this new field used biological information and gained a new profound view of how biological systems work. Molecular biologists asked new types of questions that would be inconceivable to the older generation of researchers, such as how cellular machines converted inherited biological information into functional molecules like proteins. So this has been a question, of course, that's not only relevant to evolutionary biology, but really goes back to the sort of the early days of thinking about DNA as a helical structure, as something that, basically is encoding things for proteins to express and to turn into function. So there's always been this link between function and sort of the molecular milieu of an organism. How does that molecular milieu turn into the phenotype? How does it do things like metabolism and so forth? But then there's always been this idea of function in biology. And some of that goes back sort of to the pre-scientific era, where we thought about function very much in a non-evolutionary sense. So like functions can be things that do something that confer some sort of advantage or trait. And so we kind of, when we think about function, and sometimes we think in functional terms, like anything that exists has to have some use. And so functional thinking is very seductive and convenient for thinking about an order, a sort of a natural order, or sort of an order of how things should be. But of course, just thinking about it in that way isn't enough. We need to find the mechanism. We also need to figure out whether something actually is functional, or if we're just ascribing a function that is spurious. In other words, like thinking about door handles, and thinking about those are obviously have some sort of function because humans designed them for a function on things like doors or cars or whatever. But if you see something that looks like a door handle in nature, it doesn't mean that it was put there by some sort of creation process, or even by some sort of evolutionary process. It just means that it has this shape, and indeed things like that might end up evolving into something with a function, or it might have no function at all. And we can't know until we find the mechanism, until we figure out the evolutionary history of this thing. And in fact, it's better to assume that things have no function at all than to assume that everything has a function. So this is the world that we live in, with molecular biology, but also biology more generally, evolutionary biology, etc. And so we have this collision of sort of modern techniques with sort of remnants of sort of pre-scientific philosophy coming together to think when we, influencing how we think about function and how we think about evolution. So because we were able to discover the structure of DNA, we were able to sort of ask new types of questions. And some of those questions involve things like how cellular machineries convert inherited biological information into functional molecules like proteins. The new focus on biological information also gave molecular biologists a way to link their findings to concepts developed by genetics and the modern synthesis. Now, when they talk about biological information, they're really referring to the base pairs on a backbone of a helix, and then converting that into a set of RNAs, and then translating that into proteins, into amino acid sequences that assemble into proteins. So that information isn't, the kind of information we might have in a book. People have tried to make these kind of analogies, but it's really that you have specific combinations of base pairs that translate into specific amino acids that contribute to specific protein structures and secondary structures as well. And so we have this information that gets transferred. So it's specific sets of, I don't want to call them instructions, but basically they're specific pieces of information, binary things that get transmitted and define things to look like one thing instead of another. And the reason I'm hedging here is because there's this temptation to, again, go into functional mode and say, well, there's a code here that we can uncover, and that it's more profound than maybe it is otherwise. And so that's what I want to avoid. And that's kind of what we're getting at with using neutral theory, because the argument here is that a lot of this kind of thinking about DNA, and when you have a structure to how DNA is organized and how RNA is organized, that we kind of think of this in terms of being organized in this functional way. And that's not exactly the way we want to think about it. The authors here think that, if you don't use something like neutral theory, that we go astray and we kind of go off into these fields of conjecture. So this new focus on biological information also gave molecular biologists a way to link their findings to concepts developed by genetics and the evolutionary modern synthesis, which of course integrated Darwin's thinking about natural selection with population genetics, and gave us mathematical tools to look at that, to look at the changing allele frequencies over time. However, by the late 1960s, this all changed. Elevated rates of mutation, unsustainable genetic loads, and high levels of variation in populations challenged Darwinian evolution a central tenet of the modern synthesis, where adaptation was the main driver of evolutionary change. So this is thinking about, when they say Darwinian evolution, I guess what they're getting at is like that things are all the product of selection. So again, we have this functional aspect where, we assume that something exists because it was selected for, because it had a function, and that function was optimized through selection. And what began to find in evolution or in molecular biology was that you had these things that spoke to things that weren't, selected, optimized through selection. So you have all sorts of, especially genetic load and thinking about variation, which doesn't necessarily map to the strict functionalist view. I'm not crazy about the language. I don't think Darwinian is like kind of the right way to think about it. But in any case, this is a challenge of the modern synthesis where we don't want to, there's this tendency to think as adaptationists, but of course there's something else there, which is of course where Muthu Kimura comes in, and he developed this neutral theory of molecular evolution. So instead of thinking of everything as sort of the product of selection and thus being functional, he thought of things as being neutral. Most mutations, for example, were neutral with respect to evolution. That you had genetic drift, which of course was where a population was subsampled, and whatever allele frequencies were in that subsampling of the population became dominant in that small population and persisted in the absence, a lot of times, of selection. So these are things that, of course, exist in nature, and we have to explain them. And so Muthu Kimura advanced the neutral theory of molecular evolution, which advocates that selection in multicellular eukaryotes is weak, and that most genomic changes are neutral and due to random drift. So again, random drift is acting as a force of evolution, pushing subpopulations away from that equilibrium, doing the same work as selection might do, but not having the same sort of functional consequence as selection. Also, the kind of the point here is that selection is actually weak in a lot of multicellular eukaryotes. So we shouldn't assume that selection is even particularly strong when we do see things like adaptive changes. And so this is, again, something that was proposed in the 1960s. It was elaborated on by King and Jukes, Jack King and Thomas Jukes, in 1975 in their paper Non-Darwinian Evolution, where they point out that the observed changes seen in proteins and the types of polymorphisms observed in populations only become understandable when we take into account biochemistry and this new theory of neutral theory. And so we can't necessarily say that, we can look at like phenotypes and we can assume function as a consequence of selection, and it makes a lot of sense, right? You have functions, you see a function that's been selected for, you see a function that's well adapted to its environment, you assume the processes is that this is all the product of a tight feedback loop of natural selection and function. So everything is kind of in place, and it's, I guess it's hermetically sealed in the sense that it's kind of, we have, say, like hands for grasping things, and those hands, of course, evolved to grasp things. And so there's no kind of room for molecular stochasticity there, because you trace it back maybe to genes for building a hand for grasping, and you can map those genes to the phenotype, and everything is, makes sense. But of course, what they're arguing here is at the molecular level, at least, there's a lot of stochasticity. There's a lot of data that suggests that things don't evolve in this tight feedback loop, that you have changes that are atypical of a tight feedback between selection and function. And so that's how they're describing sort of the world of the molecular milieu of neutral theory. So these were two important works, the Mutu Kimura work in the 1960s and King and Jukes. That's sort of established neutral theory as, I wouldn't say an alternative to the synthesis, but it kind of got incorporated into the synthesis a little bit. And then, it's a biochemistry theory, of course, but it's something that is, it's not something that really challenges evolution. It's just something that we also need to take into account. So it doesn't really fall into the category of the expanded synthesis either. It's just kind of in between. And so this is something that we need to kind of incorporate into I think, better into our theory and how it fits into our concepts of function. And they talk about the ENCODE study in this paper. They don't really they kind of broached the topic, but this was a huge thing back maybe about 10 or 12 years ago, where the ENCODE consortium that released a bunch of papers that made the claim that genomes were like 80% functional. And there were a lot of people who pushed back on that, and something that was called the function wars, where they said, well, you can find from bioinformatics and genome sequencing, you can do these association studies with function, and you can find very high levels of function. But neutral theory doesn't predict that. And in any case, we can show that most of the genome is junk. So there's this debate between certain groups as to how much of the genome is functional. And it ties directly back to this neutral theory. So 50 years later, most molecular biologists remain unaware of these fundamental advances. Their adaptationist viewpoint fails to explain data collected from new powerful technologies, which can detect exceedingly rare biochemical events. For example, high-throughput sequencing routinely detects RNA transcripts being produced from almost the entire genome, yet are present in less than 1 copy per 1,000 cells and appear to lack any function. So, like the ENCODE study, where they did these, used these new technologies to say that the genome was 80% functional, we can also use technologies to show that the stoichiometry of rare events, or things like, if you transcribe a gene once every 1,000 in one in every 1,000 cells, that you have processes that produce things at a very low stoichiometry that are non-functional, and if you add those together, that adds up to a large proportion of the genome. So there are a lot of ways to look at like biological processes and how they contribute to the variation that we see, and then trying to sort of classify that into function becomes hard, because these things are very rare, these events are very rare, and they have a very low sort of copy number, have a very low stoichiometry in the cell. When I say stoichiometry, I just mean the amount, the degree of which a molecule is expressed. So sometimes if it's a very low stoichiometry, you have, a lot of, it can vary by its sort of stochastic nature. So you have one event that can produce 5 molecules, you have another event that can produce 1 molecule, and you have a lot of fluctuation there that is not in line with a very refined function, or any function at all for that matter. So there are all sorts of things like that in biochemistry. We can uncover those with newer tools, and it sort of supports this idea that neutral theory explains the data better. So, this is something, then they talk about how molecular biologists kind of, do a lot of experiments but don't have a guiding theory, and this is the guiding theory that they propose. But molecular biologists must now reincorporate ideas from classical biochemistry and absorb modern concepts from molecular evolution to craft a new lens through which they can evaluate the functionality of transcriptional units and make sense of our messy, intricate, and complicated genome. So a few highlights from the paper. So the paper kind of lays out a nice history of evolutionary theory with respect to a lot of biochemical findings, and how a lot of the early biochemical work really contributed to sort of a challenge to kind of what we thought of as conventional evolutionary theory, at least looking down from the phenotype and thinking about what's going on at the molecular level. So one finding is, of course, of the molecular clock. So there's this idea of the molecular clock where, if you look at mutations in different lineages, you have, the assumption, of course, in descent from common ancestry would be that those changes not only sort of share a common origin, but should mutate at a similar rate because they're from a common ancestor. They should share that evolutionary rate, and they should share mutations, or they should share sort of they should share an underlying mutational structure. And so in 1962, it was found that the number of amino acid differences between the hemoglobin proteins isolated from any two species correlated with how long ago they had diverged from a common ancestor. And this was, of course, based on paleontological estimates. So you can think about like how long ago the common ancestor existed between any two taxa, and you can kind of say, okay, we should see a certain number of amino acid differences if there's a common rate of mutation, and that's, of course, exactly what they found. And this is this concept of the molecular clock, which is often used to calibrate phylogenies. And so this is an important finding that said that basically the different proteins and protein paralogs evolve at a constant rate in different lineages. And so that was the first kind of finding in molecular biology that related to evolution, and it kind of suggested that there was this conservation of things in terms of mutation. The second part here is that Jack Hubby and Richard Lewontin found that different individuals within a species had a surprisingly high amount of variability for any given protein. So not only did we have this molecular clock, but we also had a lot of variation in how proteins were expressed across the species. So you had this intraspecific variation, and you had this interspecific regularity. And so it's just really interesting how these things deviated from the population genetics models that had been proposed back in like the 30s and 40s. So others noted that many of these substitutions led to a mostly neutral change in proteins, which means that a lot of these mutations, a lot of this variability, didn't really mean anything in terms of evolution. It was just change in the protein sequence and not necessarily corresponding change in function. And so this is when we get to Muthu Kimura's 1968 publication, Evolutionary Rate at the Molecular Level. And so in this paper, Kimura finds that the amount of protein variation between and within species, estimated by the new molecular biology techniques, inferred such a high substitution rate that if they all consisted of alleles that were under selection, the cost of replacing these alleles would be intolerable. And he uses the term genetic deaths. So this is where alleles would have to be sort of selected out of the population. And so the idea is that there's this, that basically if you have this that given the amount of protein variation, if we assume that there's some function to it, that you would have to keep killing off alleles, and it would sort of undermine the entire concept of sort of functional improvement by genetic change. Basically, there wouldn't be enough genetic material around to facilitate evolution as proposed. where natural selection was proposed. So Kimura proposed instead that the majority of these mutations must be neutral. Furthermore, Kimura demonstrated that slightly deleterious or beneficial mutations behave like neutral mutations, provided the absolute value of their selection coefficient was smaller than the inverse of the effective population size, which is here. So basically, that in cases where selection isn't absolute or isn't very high, you have these neutral processes that dominate. So in this equation here, we see the absolute value of S is smaller than this 1 over the effective population size. The selection coefficient being S is the average decrease or surplus in offspring in the mutant compared to a wild-type individual, and the effective population size is the number of breeding individuals that randomly mate in a species. So this is where we have to consider population size. We have to consider the decrease or surplus in offspring in a mutant population, or in a population where those molecular changes have occurred, and then we take the proportion of that. In practice, the effective population size is much lower than the actual population size, as it is dependent on a number of different parameters. In humans, the effective population size is estimated to be about 10,000, which is a bit lower than the typical number for most other animals. If a mutation falls within the parameters of the above equation, it does not clear what Michael Lynch calls the drift barrier, and its fixation will be dictated by neutral evolution. So we have this idea that neutral evolution is can dominate when selection doesn't dominate. So when selection is weak, or when selection is sort of moderate, we can have these neutral processes, and they can dominate what we see as evolutionary change. And a lot of that neutral change is not necessarily functional. A year after Kimura's paper, Jack King and Thomas Jukes independently established a similar conclusion. This is King and Jukes in 1969. However, many of their arguments relied on the biochemistry of proteins and the types of mutations seen in nature. And so there's this debate in evolutionary biology, the neutralist selectionist debate. Some population geneticists in the selection camp have advocated that most mutations can reach fixation because they are linked to nearby alleles that are under positive selection. This is evolution by drift. And then these arguments do not invalidate the idea that most mutations by themselves are nearly neutral. The main thesis of the neutralist camp, indeed, as pointed out by several commentators, rampant draft, if anything, lowers the effective population size and would thus further erode natural selection's ability to weed out slightly deleterious mutations. So, although many details about molecular evolution are still debated, the main tenets of the neutral theory have been confirmed by numerous studies. Predictions made by the neutral theory match closely what is seen in countless analyses of single nucleotide polymorphism distributions, or SNP distributions, and comparative genomic surveys. And then there's some references given there. So one of the take-home messages of this paper is that molecular biology is drowning in a sea of data and needs a sort of a unifying theory or a form of knowledge that can unify the field. So another set of papers I'm going to review maybe next week or another time is this theme of shape changes in soft materials. So this is a multiphysics-based concept where they're using multiphysics, and of course we talked about multiphysics last year, talking about different aspects of the physics contributing to shape some phenomena. So we're interested in biomultiphysics, which is how biological soft materials are formed by these different physical processes and how you can model those processes all at the same time. And so this is a paper on multiphysics-based control shape change in soft materials. So I did a literature search on this, and I found that there's a lot of stuff on multiphysics and soft materials, but not a lot in terms of how that affects sort of biological tissues and developmental biology, which is, of course, our interest. So I have a few references here that kind of go through this idea. One of these is from Frontiers in Materials, and this is an editorial on tackling the multi-scale phenomena of soft materials for engineering innovations. So this talks about the promises and challenges of soft materials, the role of multi-scale understanding, which is a key, of course, not only for just generic soft materials, but for biological systems as well. Different types of initiatives, applications in engineering, and the need for mechanistic studies. And all those, of course, we know about from our work in the group on tensegrities. So there's basically in this editorial, they talk about the study of soft materials being a cornerstone of materials science. It's advancing what they call the materials 4.0 era, which we're, I guess, apparently in, which is expected to drive the fourth industrial revolution. There are also, furthermore, initiatives like the Materials Genome Initiative that are at the forefront of addressing critical material challenges. Soft materials have diverse and complex properties, ranging from amorphous and hierarchical structures, or ranging from amorphous structures to hierarchical structures, and hold immense innovative potential. Now, their full exploitation is hindered by an elusive process-structure-property relationship, which remains a significant barrier to their application across various engineering fields. Despite this, advances in computational capabilities and experimental techniques are paving the way for breakthroughs. So there is a lot of promise, but there are also a lot of challenges. This process-structure-property relationship is something I think they're arguing can be understood through multi-scale, like understanding soft materials and the multi-physics of soft materials as this multi-scale endeavor, and certainly in biology, that's sort of the key aspect of this. So, if we have, say, like a tissue, the tissue is made of cells, the cells express proteins, which are expressed by genes, and we need to understand how all of those work together. So the process structure property relationship is central to materials science, yet soft materials remain poorly understood. The relationship governs how the processing of a material influences its structure, and how that structure in turn determines its properties. So, yeah, for soft materials, we have different phenomena that operate across different scales, and it makes it really hard to figure out this relationship. And so this is similar to the idea that we talked about earlier about function, and how function is, instead of function here, we're talking about structure and function. So, this is, again, a very similar problem. How do we map what we're doing in our experimental work to this concept? And maybe it's an outdated concept. Maybe we need to rework our concepts a bit. But that's kind of what they're getting at here. There's a lot of interplay between different processes. Things that happen at the nanoscale, for example, can significantly influence its macroscopic properties, but it's hard to sort of map that in a linear fashion from one scale to another. And so this is why people are turning to multi-scale modeling and simulation, which allows for examination of material behaviors across different scales. And so then, of course, the obligatory mention of AI and machine learning methods and how those can be helpful. But I think also good conceptualization and good models for transferring between scales is also important. And then finally, this need for mechanistic studies and how we can use those mechanistic studies to sharpen our understanding of these phenomena and how they interact. This paper, which is an abstract, it talks about multiphysics-based controlled shape change in soft materials. So here again, we have this idea of this idea of using multiphysics as a way to understand these hierarchical soft materials. So what they're trying to do is they're trying to control shape change. So a precise controlled shape change of responsive materials requires the development of theoretical and computational approaches capable of reproducing the involved physical phenomena and their interaction. When polymer-based materials are considered, shape change capabilities can be obtained from external stimuli such as temperature, light, humidity, electrical field, and many other things that can affect this polymer. And so we have these inputs that can trigger shape change. These stimuli are precisely spatiotemporally controlled. In other words, if we can control where they affect the polymer, what location and at what time, can deform a structure into the desired configuration at a given time. So we want to, say we take a developmental process, for example. We want to deform a certain part of an embryo at a certain time. We have to know kind of what that underlying process is and when some stimulus of whatever type needs to be delivered at the precise time. And so this, in development, we're also concerned about stimuli which are molecular. So there might be some molecular cascade that's that's activated, or some sort of a bioelectric signal that's activated, but the principle is the same. This paper investigates the multiphysics-based driven shape change in highly deformable composite structures made of a passive elastic structure and an active one capable of responding to an environmental stimuli. So this is where we have a passive elastic structure and an active structure, an active elastic structure, and we look at these two different forms that are both triggered by environmental stimuli. By harnessing different mechanisms, which are these 4 mechanisms. So the first is the swelling of gels. The second is deformation of electroactive dielectric elastomers, or DEs. The first one is the swelling of gels and the deformation of electroactive dielectric elastomers, or DEs. The second are controlled mechanical stresses that can be transmitted to the passive portion, leading to its deformation. And this is done in the present study by an elastic tube filled with a gel, where the volume deformation is driven by the fluid uptake. 3 is a bilayer structure made of an elastic substance and a DE, or dielectric elastomer, active layer, whose deformation is induced by an electrical potential. And 4, where these are considered as morphing structures. So we go from the swelling of gels, the deformation of some sort of dielectric elastomer. There are controlled mechanical stresses that can be transmitted. This is done through an elastic tube filled with the gel. Then we have, of course, the bilayer structure. And then 4 is where they have these morphing structures. So it is shown that by properly distributing the mechanical properties in this hyperelastic passive element, interacting with the responsive active material, precise shape change can be achieved. To this end, we numerically solve the multiphysics coupled problems and adopt a machine learning approach for solving the inverse problem. Given a prescribed target shape, the proposed approach enables us to obtain the proper spatial stiffness distribution of the passive electric element, enabling the achievement of the desired deformed configuration. As you can see, that you have a couple of different types of physics here. We have electrical physics, we have, of course, mechanics, we have this elasticity, and then we have these different sorts of the physics of layers and the elasticity that we have to account for. So we have all these things working together to form these morphing structures. So again, we're just trying to change the shape at a certain time in a certain location in the structure. And so we can map this to, say, like soft robots or other types of applications where we're interested in this sort of morphing. But we can be more daring and think of biological systems and sort of poking biological systems from the phenotype level. And so what's interesting is when you start to add in real biological systems, because of course real biological systems have a lot of things underneath the hood, a lot of molecular pathways, and using those molecular pathways as things that can trigger changes as well, be maybe very interesting, but maybe also make things even more complex.